Hi, my name is Justin Gaudon, and I'm a postdoctoral research fellow at the University of Waterloo and Rare Charitable Research Reserve. I was asked to talk a bit about my research at Rare and some of what I've been working on for the past year and a half. My main research project right now focuses on the biodiversity and conservation of beneficial insects. Understanding the relationship between beneficial insects, so bees, predatory wasps, parasitoid wasps, and vegetation may help us identify plants and habitat to use in site restoration for the purpose of maintaining or increasing populations of beneficial insects at Rare. The specific plants and habitat within a site might serve as refuge or food and attract specific kinds of helpful insects. The presence of these beneficial insects may then in turn provide really important ecosystem services such as slowing or reducing the impact of insect pests or increasing pollination. Instead of looking for the insects themselves, which is a lot of work, it requires expert knowledge and equipment like really high-powered microscopes to identify many of the really small species. It might be much faster and easier to identify certain plants and habitat that are associated with the presence and certain numbers of these beneficial insects. Incorporating strategies that integrate beneficial insects into planning efforts by identifying and planting associated vegetation could be a very practical solution for restoration ecologists and other landowners and conserving populations of beneficial insects at Rare. To investigate this, we set up 21 Malaise traps across the Rare Reserve. And if you don't know what a Malaise trap is, it's a large tent-like structure that's intended to capture insects as they're flying. And the insects get trapped in the tent, and then they move up to the sunlight that's coming in at the top of the trap. And then they enter a collection jar and can be used as a sample for the insects in the site. And what we did was we installed the traps in different habitat types, and we identified those habitat types through ecological land classification. So we knew, okay, this area is a sugar maple oak deciduous forest, or this area is a yellow birch deciduous swamp, or a naturalized coniferous plantation. And then we used a vegetation sampling protocol to identify all of the plants and habitat characteristics to really get a complete picture of the vegetation within 400 meters squared of each trap. We set up the traps in May last year and left them up until August, and we serviced them every 7 days or so. So we went and we collected the insects from the collection jars and replaced the collection jars and replaced any damaged traps. From these samples, all of the hymenopteran insects, so those beneficial bees and wasps, were identified and sorted from the larger Malaise samples, which contain thousands of insects. And in July 2020, we sent just shy of 27,000 hymenopteran insects to the Center for Biodiversity Genomics at the University of Guelph for bulk DNA barcoding analysis. And we received the results of the analysis back in August 2020, so not too long ago from when I'm recording this video. And right now I'm going through and processing the data that we received to compare the insect communities and look at how they change across different vegetation communities to determine if plants and habitat can be used to predict beneficial insect abundance and diversity. And so from this work, we're hoping to establish a strong basis for considering beneficial insects when creating and improving ecological restoration and conservation planning programs at Rare. So if anybody has any questions, feel free to reach out to me, and I'd be happy to chat about some of the work that I've been doing. Thanks.